everyone, and welcome back to the channel. So in this video, I'm going to go through genetic screens. So genetic screens are things that are typically done with genetic model organisms, things like yeast, fruit flies, C. elegans. And the purpose of a genetic screen is to either identify genes that produce a given phenotype or perform a screen to find out which particular genes are causing a particular phenotype. And then we might also do follow-up screens called enhancer screens or suppressor screens to find other genes that are either involved in the same genetic pathway or genes that might be in parallel pathways that function to either oppose a particular phenotype or contribute to a given phenotype. So a lot of this sounds confusing, but let's start by just introducing ourselves to what a genetic screen actually looks like. So there are two different types of genetic screens. There are forward genetic screens and reverse genetic screens. So when we talk about forward genetics, this is referring to what is pretty much the traditional approach. In other words, you isolate or introduce some type of mutation, you examine the phenotype, and then you do what you can to identify the gene in which the mutation is located, right? So you do something. Right. Or perhaps you're looking at a group of flies or a group of worms or a group of yeast, and you say, oh, that's interesting. That's something I've never seen before, or that's a phenotype that I'm interested in. I wonder what mutation caused that, right? Because it's something that deviates from the wild type. So you might think, okay, well, I've got this cool little mutation. I want to know more about it. Let's see if we can figure out the gene in which the mutation is located, right? Alternatively, you might be doing a forward genetic screen where you do mutagenesis. You do something to induce mutations and look for a specific phenotype. Okay. Now, the reverse genetics is the opposite of that. You take a gene that you are interested in, you do something to either mutate or inactivate a particular gene, and then you examine the phenotypic consequences of that. So in forward genetics, you start with the mutation and try to identify the gene. In reverse genetics, we start with the gene we care about, and then we try to mutate and inactivate it and find out what happens as a result of that inactivation or of that mutation. Okay. So let's talk a little bit about forward genetic screens. So forward genetic screens, typically you're going to induce mutations through some type of mutagenic approach. All right, this is also known as a mutagenesis screen. So you can do that chemically with certain chemicals. You can do that by exposing animals to UV light, which is known to induce mutations in DNA. And typically when we're doing a forward genetic screen, we're going to be working with some type of model organism that is relatively easy to work with. So Saccharomyces cerevisiae, which is yeast, Drosophila melanogaster, which is fruit flies, C. elegans, is another great example, which is an nematode worm. You also have Arabidopsis and the bacterium Escherichia coli. And then to mutate these, you might use radiation such as UV light, you might use X-rays, or we use chemical mutagens such as ethylmethane sulfonate, EMS, or N-ethyl N-nitrosourea, ENU. These are commonly chemicals that are typically used to induce mutagenesis. And what I want to state is which mutagen you use, how you utilize that to induce mutations. It really depends on which organism you're working with. For example, with C. elegans, you can typically just expose them to these chemicals when you have a gravid female. It's a female loaded with eggs, and those mutagens can impact both the adult as well as the eggs that are inside of the hermaphrodite. They are egg-laying hermaphrodites. That's just a characteristic of C. elegans. So when we do genetic screens, we're typically looking for some type of easy to observe phenotype. This could be some type of change in movement. It could be some type of morphological defect. So for example, here you can see you've got problems with the wings. You could be looking for some type of developmental defect, changes to the developmental process. It could be metabolic, where you're looking at which foods they can use, which foods they can no longer use. So this is a yeast assay that you're looking at here. It could have to do with aging. So C. elegans is a very commonly used organism for looking for for age-related defects. It could be defects in tumorigenesis. It could be to do with fertility and fecundity. So how much offspring do you produce? It could be cell cycle deficits. It could be the ability to detect certain sensations, whether it's touch or olfactory, or you could be looking for a particular disease phenotype. I mean, really, it's limitless, but these are some of the most common phenotypes that people perform forward genetic screens to identify. So the way forward genetic screens were typically done in the past was through linkage analysis, right? So we had these spontaneous mutations in certain species that we were able to map. I've talked about linkage analysis and how this was used to map chromosomes in the past in one of my previous videos. But as we got into the 80s and 90s, short nucleotide polymorphisms or SNP mapping and positional cloning took over. That allowed us to be more targeted with our genetic screens. It made it easier to identify which genes were mutated in one of these screens. And now with modern approaches, it's much easier to do the sequencing to figure out where those mutations happen to be. One of the really neat things that's come about, actually, there was a Nobel Prize awarded for this in the, I want to say it was in, I can't remember what year, but it was in the late aughts or the early 2010s for the discovery of something called RNA interference. It was discovered in the nematode model organism Caenorhabditis elegans, but it's since become it's now known that RNAi is much more widespread. It's a phenomenon that happens in most eukaryotic species. But in worms, it's very, very easy because you can quite literally knock down genes by using targeted RNAi, either through microinjection, soaking them in the RNAi, feeding it to them, or expressing it as a transgene. And basically, by providing them with these interfering RNAs, you can really severely knock down the expression of a targeted gene as long as you know the sequence of the gene. And for the record, the C. elegans genome is fully sequenced and annotated, so you shouldn't have trouble identifying the sequence of a gene, and then providing it with RNAi against that gene, and you can actually observe very specific phenotypes. By knocking down those genes, you can greatly reduce the expression of certain genes by providing them with the RNA against them, which makes it very, very easy to observe a mutational phenotype in these animals. Just because you're just like, hey, here's the RNA, eat this for a couple generations, and now we'll look at the phenotype and see if there is one. So you don't have to go through the process of doing these randomized mutagenic screens and hoping for the phenotype you're looking for. You can literally go in and say, this is the gene I'm interested in. Let's see what happens. That brings us to reverse genetic screens. So in reverse genetic screens, we are going to begin with a previously identified gene. All right. So there is a gene we are interested in. Maybe it is the homolog to a gene in humans that causes a disease. And we want to know what happens if we alter that gene's expression or knock it out entirely. But when you do reverse genetic screens, the thing that's made this much more possible is our ability to induce very targeted mutations. I just talked about how we can use gene silencing, so RNA interference or short interfering RNAs. As long as you know the sequence of the gene, you should be able to knock it down in pretty much any cell you want to using RNAi or siRNA, depending on which organism you're working in. We've also gotten a lot better at doing site-directed mutagenesis, which means we can target specific genes. We can do gene targeting, which allows us to do knockouts or knock-ins. So you could knock a gene out. You can introduce a gene that doesn't normally exist in that particular organism to see what happens. We also have the ability to create transgenes. I talked about this in my previous in a previous video of mine where I talked about genomic techniques. One of the things you can do is create transgenes, and you can actually overexpress a particular gene so that it's present in higher amounts. So you can examine not only what happens when you knock that gene completely out, what happens if you overexpress it, right? What happens if you produce too much of this? Do you get the opposite phenotype? Okay. And the question we're usually seeking to answer is, what are the phenotypic consequences of the loss or overexpression of a gene? So, for example, we might be asking, Does loss of function or gain of function, LOF-GOF, in this gene mimic a phenotype similar in humans, right? We know people who are missing this gene or have a mutation that interferes with the function of this particular protein have this phenotype. Can we recapitulate that phenotype in my organism? Or maybe we know that like if this gene gets overexpressed in humans, it causes tumors. Does it do the same thing in mice? Does it do the same thing in fruit flies? Okay. What processes or pathways is the gene product required in? Right. So, this particular gene in humans is involved in insulin signaling. Is what we think to be the homolog in C. elegans doing the same thing? Does it have similar phenotypes? Is it involved in the same pathway? Who does it interact with? All right. And again, does this behave in a manner similar to potentially related genes in other organisms? Cool. We found this neat thing in fruit flies. Does it do the same thing in C. elegans? Does it do the same thing in mice? That might provide us information of this is a conserved effect. So is this something that we can explore as a potential therapy in humans? Or is this something that could be causing certain diseases in humans? Now, a lot of what I'm going to talk about here in terms of these sort of knock-ins and knockout strains, a lot of this is being supplanted by a newer technology called CRISPR. But I'll give you some information about how this is done in mammals. So Cre-LoxP is an older system, still used heavily, and still very, very reliable. And in fact, they're kind of taking this particular system and merging it with the CRISPR-Cas9 system that I was just referencing. But it's used to create transgenic mouse strains. First you introduce a transgene called Cre-recombinase, and then you express a transgene that is shouldered by sites that these enzymes can work on. And then basically when you induce the expression of Cre-recombinase, it goes in, it removes these roadblocks, and all of a sudden, boom, that gene is now being expressed. And you can induce these by mating the Cre mouse with the floxed mouse with this LoxP gene. You can end up getting expression of a particular transgene in mice, which can be very challenging in other cases. All right, this is producing what we call a knock-in, or this is a knockout. Here they're knocking out the targeted gene. You can do the same thing to do a knock-in to overexpress a gene or express a gene that's not supposed to be there. All right. And again, this has been merged with the CRISPR-Cas9 system because the CRISPR-Cas9 system is very, very good at introducing targeted mutations or targeted expression of transgenes in organisms, which is really cool. And I'll have another video about that one soon. In terms of C. elegans, or C. elegans and other organisms, RNAi, so RNA interference or short interfering RNAs, siRNA can be used. I discussed before how C. elegans is an incredible system for doing this because there are so many ways to do RNAi soaking, feeding, injection. In Drosophila, it's a little bit trickier. Typically, you have to soak cells in culture. So if you're doing fly culture, you can do it this way, but it's hard to do it in the whole organism. If you want to do it in the whole organism, you have to micro-inject into embryos, which sounds tricky, but actually after you've done it for a while, it's actually pretty easy. And then when we talk about mammals, quite often we have to do siRNA in cell culture. The only other option would be to create a transgene expressing the siRNA and then induce its expression in a living organism, which is trickier than it sounds. So microinjection sounds tricky, but it's not that bad. Transgenic expression sounds easy, but it's actually kind of challenging in mammals. So the other thing we can do is we can express gain of function mutations. We can do this by creating strains that carry extra copies or gain of function alleles. Often we tag these with fluorescent proteins. So here you can see C. elegans, you can see that they've knocked in this particular gene. It's being overexpressed, and you can see it goes to the nucleus. That's why these little nuclei are all lit up like that. We can also overexpress by doing transfections into mammalian cells. We can inject transgenic arrays into fruit flies and C. elegans. And again, CRISPR is starting to get used so that we can do site-directed mutagenesis. So lots of different reverse genetic techniques that we can apply here. So these are great. Both forward and reverse genetic screens are great for either identifying novel mutations that cause desired phenotypes or looking at a gene and discovering what the consequences of its loss of function or gain of function can be. But what if we want to know more? What if we have a cool phenotype or we have a gene that we're very interested in, but we want to know more about which pathways these genes are involved in or who they play with? Like what other genes, what other proteins do they interact with? And this is where we can do enhancer screens or suppressor screens. So when we talk about enhancer screens, we need to start first with an identified mutant strain. And just to be clear, doing this with RNAi, starting with an RNAi treated strain to do an enhancer or suppressor screen is not generally recommended, because remember that RNAi is only knocking down the expression of a gene temporarily. And it's hard to do that type of analysis with RNAi. But sometimes we have done it with double RNAi. Just realize that those results need to be followed up, usually using true mutations at some point. Okay, so we begin with an identified mutant strain, and then we search for additional mutations in other genes that produce an additive phenotype. All right. So what I'm saying by that is, let's say you have a mutation, and we'll use worms as an example because that's where my expertise lies. You have this mutant that lives twice as long as a wild type animal. All right. Normal worms live about 21 days. These guys live like 40 days. And you're like, wow, that's really cool. Well, I need to find out when I need to figure out if there are other pathways that are involved. So how do I do this? Well, what I do is I take that mutant and then I do another mutational screen using, mutational analysis. In this case, we started with a mutant. We can use RNAi to look for enhancers, and we're looking for animals that live even longer, right? Because that would help us find out that, okay, well, it's not in the same genetic pathway, because if it was in the same genetic pathway, we would expect them to cancel out, right? If I mutate this pathway, it shouldn't matter whether I mutated at one place or two places. This pathway doesn't work. So if I get a phenotype for mutating this pathway, and then an additive phenotype from another mutant, it argues that it's probably in a parallel pathway that does the same thing to influence aging, or that does something different to influence aging. Okay. The other thing we can do with enhancer screens is we can start with synthetic mutants that only show a phenotype when both genes are mutated. So sometimes we can have a mutation and you're like, man, like I really thought that mutating this particular gene was going to have an impact. And you find out that it doesn't. So you take that mutant, you then you do another screen to look for subsequent mutations, or use RNAi or siRNA to see if, okay, well, this gene is mutated and it's not doing anything. Can I get the phenotype I expect by finding one of these other genes? And perhaps what's happening is you've got parallel pathways that are a bit redundant. So when you shut down this pathway just picks up the pace. So you have to knock out this pathway too to get the phenotype. So either way, when we do enhancer screens, we are looking for genes that either work in the same pathway as each other in an additive manner or operate in parallel pathways to influence that phenotype, i.e. they act redundantly. So that's what enhancer screens are going to be doing. And you can see kind of what these outputs look like, okay? To figure out whether they're in the same pathway or in parallel pathways, you're going to do a little bit more work, all right? So again, here are some examples of this. I talked about longevity. So one of the things I can tell you is that EAT2, this mutant lives, here's your about 21 days for median lifespan here. We're right around 30 days for median lifespan in EAT2 mutants. If we look at DAF2 mutants, a totally separate pathway, we see that, okay, when we reduce the expression of DAF2, now they live 40 days. So for wild type, median lifespan about 20 days. For EAT2 mutants, median lifespan about 30 days. And for DAF2 mutants, the median lifespan is about 40 days, right? So with DAF2, we get a full doubling of longevity in these animals, which is really, really cool. But look what happens when we take the EAT2 DAF2 double mutant. Now our median lifespan is out to like 55 days. And this work has been done repeatedly. And one of the things we know is that DAF2 and EAT2 are in completely separate parallel pathways that both impact lifespan. Here's another example over here. Here we have these are yeast strains. So this one is missing a protein or a gene called URA4. This one is missing a gene called SWI3. And what you're seeing here is, depending on so over here, these green, yellow, red, and blue boxes represent subsequent mutations. And you can see that these 4 really don't have any impact on the URA4 mutant. There might be a small growth deficit here with DDB1, but let's look at SWI3, and you can see there's almost a complete inability of these organisms to grow. So you can see that this phenotype has now been enhanced. It would argue that these 4 genes are not in the same pathway with SWI3. They operate in a separate pathway to control the growth phenotype, whereas they're probably within the same pathway as URA4 because they don't add to the particular phenotype. Suppressor screens are the exact opposite, okay? Again, depending on what we're looking at, we can find genes that work in the same pathway. We can also find genes that operate in parallel pathways, depending on how we look at this. So, for example, again, we're going to start with an identified mutant strain, and we're going to look for genes that reverse the phenotype, okay? So this particular growth defect actually results in the worm producing additional sex organs. So this organ right here is called the vulva. This is where the eggs come out. Typically, C. elegans hermaphrodites have one of them. It's located right there. But you can see in the Lin53, Lin15A double mutant that they have what we call the MUV phenotype, the multiple vulva phenotype. They have 3 of them. Now, these 2 are not really functional, but you can see the growth of the cells that represents an additional vulva here, actually 2 additional vulvae here, or vulvas. I'm not actually sure of the plural of that because there's only supposed to be 1. But when we introduce this new mutation, ISW1, the phenotype is completely rescued, which means that whatever Lin53 and Lin15A do, ISW opposes it because by antagonizing this double mutant with the ISW1 mutant, we rescue the phenotype. This is a suppressor. Why? Because this particular mutation suppresses the mutant phenotype of the double mutant. So what I wanted to do in this video is give you a brief overview about genetic screens. Genetic screens are very, very interesting because they are often the beginning of a research project. I can give you a great example of this. When I was a postdoctoral researcher, so I'd completed my doctoral degree, I changed from one worm lab to another at the university. And this particular lab was interested in studying aging. And the principal investigator had just come to the university from a very prominent research institute. We'll just leave it at that. And he had performed a screen looking for mutations that impacted the longevity of a known longevity mutant. It's the DAF2 mutants that we were talking about before. And he had this list of like 60 target genes. And part of my job there was to identify several of to basically rescreen his 60 targets to make sure they were true targets and then identify some that were of interest. And that spawned a whole host of research projects within the laboratory where myself, other graduate students, lab technicians, we were all working on a cluster, our own little cluster of genes to find out how do they work within this particular organism to influence longevity. So screens are typically the beginning of a much larger research project or the beginning of several other research projects. Nevertheless, they are a great way to identify genetic targets that are of interest to you for whatever your interest is in studying, whether it's metabolism, aging, cell division. I gave you that big list of phenotypes that we might be interested in, and suppressor screens are often a great way to at least establish a baseline. Here are some genes that I should be considering if I'm interested in understanding why this phenotype happens to be. So I hope you enjoyed this video. Just a quick one to give you an idea of what genetic screens look like and how they're performed. I hope you enjoyed it, and I hope to see you again on my channel. Thanks.